Hello everyone, my name is Kefang Nie. I'm a PhD candidate in graduate group in epidemiology at the University of California, Davis. Through summer 2025, founded by Summer Immersion Program, I conducted some research and outreach for my project, from compost piles to produce safety, how composting practices influence microbial safety and food safety. Composting converts organic materials into soil amendment that supports plant growth and soil fertility. However, if pathogenic microorganisms are not sufficiently reduced during the process of composting, they may persist in finished compost and contaminate soil where produce is grown. So our study assessed microbial population in finished compost produced by different types of composting units in California, including commercial facilities, urban community composters, and vermin composting systems. We collected 184 finished compost samples from 35 composting units. We quantified fecal coliform concentration as an indicator organisms and screened for foodborne pathogens, including Salmonella, Listeria monocytogenes, Shiga toxin producing E. coli. Samples from commercial facilities generally had lower fecal coliform concentration. Less than 50% of samples exceeded the applicable regulatory threshold, suggesting the need for batch-specific process control. Targeted pathogens were not detected in the commercial facility samples. Urban and community composting samples showed higher fecal coliform concentration compared to the commercial facilities. We did observe pathogen in a small number of samples. We found Salmonella in one urban community sample, and we found Listeria monocytogenes in another 4 urban community samples. We found variation in feedstocks, which is the raw material used for composting. We also recorded a diversity of composting methods in these composting systems. We observed less consistent monitoring of the key factors, including temperature, moisture, and oxygen for composting process. All these factors likely contribute to the occurrence of foodborne pathogens and higher fecal coliform concentration that we found in urban community samples. Vermicomposting system demonstrated the highest fecal coliform concentration, while targeted pathogens were not detected in these samples. Because vermicomposting operates at a lower temperature and via a different biological mechanism, so tailored and evidence-based guidance is needed for this section of composters. We share these findings at International Association for Food Protection annual meeting and California Certified Organic Farm Field Day, engaging both academic audience and real-world practitioners, with a focus on practical, scalable improvements for facilities and community composters. To wrap up, across California, we observed distinct microbial patterns across industrial and alternative composting systems. Commercial facilities generally followed standards. Urban and community compost showed low occurrence of Salmonella and Listeria monocytogenes. Vermicomposting is small-scaled, showing higher fecal coliform concentration. Because compost is applied to agricultural soil, the takeaway is practical. Strengthen the monitoring, record-keeping, and clear scale-appropriate guidance to mitigate the microbial risk in composting. Thanks for watching. I want to take a moment to acknowledge all participated composting units, managers, lab technicians that made all this project and extension possible. For more information or questions, please contact me or Professor Alda Pilis.